Hello, everyone. What a rowdy mob. I'm Michael Farrell, and I'm the Associate Director here at Monash Biomedical Imaging. And today, it's my pleasure to introduce Thomas Andrillon, who is a relatively recent arrival here at Monash University. He's in the School of Psychology. And not only is his arrival recent, but he obtained his PhD in Paris in 2016, I believe.
Yeah.
So he's a relatively young man, or at least from my perspective he is. And despite his tender age, he's been extraordinarily productive. He's had a series of papers published in the journals under the Nature banner. And not only is he making waves on the science front, but his work's also garnering general interest in the public at large. And I'm absolutely delighted that we're going to be hearing about his work today. And so with no further ado, I'll hand over to Thomas.
Thanks. Thanks everyone for joining. I have to admit that when you research sleep and when you talk about sleep, usually it attracts a lot of attention because it's something everyone is easily interested in. It has some inconvenience as well. You cannot go to a wedding without or any kind of social event without everyone asking them about their sleep disturbances. But it's at least a research topic that connects with people. So what I will try to present here is actually some recent development in sleep research showing that sleep and wake can be often intermixed. So we can often be in state where we can be both awake and asleep. But before starting this, since I'm indeed a quite recent arrival, both in Monash and in Australia, I wanted to give a brief biography of myself. So as my accent will tell you, I'm French, but I was born here actually, which is surprisingly maybe for you French territory. And then I had the luck to spend a lot of my childhood exploring French overseas territory. And at some point, I decided that my life needed to be a bit more serious. So I went to Paris, like every French people try to do when they want to study. And I started to study biology and cognitive sciences. So I notably did my PhD with Sid Quider, who is not a sleep scientist at all. He's focusing on consciousness and development. So sleep was a new thing for him. To study sleep and how it impacts our cognition and consciousness, we paired up with both sleep specialists like Damien Léger and audition specialists like Daniel Presnitzer. I had also the opportunity to work in the United States during this period in the laboratory of Giulio Tononi and Chiara Cirelli, and with Yuval Nir, who was a postdoc at that time. Now he has his own lab in Israel. And you will see a lot of what I'm going to present is actually inspired by the work of Giulio Tononi and Chiara Cirelli on sleep and the function of sleep, the mechanism of sleep, and so on. And so I graduated from my PhD in 2016. Then I took 1 year off to travel a bit. And in 2017, I joined Australia. So working with now Suchir here at Monash Biomedical Imaging, Joel Pearson in UNSW, and we are collaborating more and more with Sean Drummond for the sleep aspects of our projects. What?
That red dot.
Which one? This one? Oh, this is Tahiti.
Oh,
Yeah.
okay.
Yeah, France is lucky to count Tahiti as one of its overseas territories. If you have the chance, I encourage you to visit. So why studying sleep? So My interest in sleep was mostly initially because it's a beautiful modulation of consciousness and the way we interact with the environment. And in many aspects, it's a puzzling one because sleep has been observed in pretty much all animal species so far. So it's a universal phenomenon in the animal kingdom. It seems a necessary one. So if you prevent animals from sleeping, they die quite quickly. So if you take humans, for example, or other mammals, you die in as many time you need to die from thirst. So sleeping in a way is as important as drinking. And despite this, it's still a risky phenomenon. So when we sleep, we lose the one advantage of being an animal, which is being animated and being able to respond to your environment. So why do we place ourselves every single night in such a risky situation? So sleep has for a long time been thought to fulfill very important functions where this state of irresponsiveness will be crucial. And that will be the reason why we transit between wake and sleep. So according to these views, then it comes almost with a direct implication that sleep and wake has to be mutually exclusive. Because if you can be in states that are mixed between wakefulness and sleep, why sleeping at all? Why putting yourself in a state of total sleep where you're not able to interact with your environment? So the issue with that view is that this notion that sleep and wake will be mutually exclusive is at odds with a lot of new evidence. And there is a lot of data, as I will present, showing that sleep and wakefulness can actually be intermingled. So that is something that was known in the literature from either sleep disorders like sleepwalking, which can be seen as an abrupt awakening from deep sleep, where your brain is in a state of mixed wake and sleep activity. But it also known that other animals can actually enter states of mixed wakefulness and sleep. So dolphin is very classical example of such states. So dolphins, being mammals, they need to breathe. To breathe, they need to go to the surface. Being close to the surface is a danger for animals. So how can you do it? How can you both sleep and breathe without putting you in danger? So this, a solution for this in dolphins is to sleep one hemisphere at a time. So when they sleep, they actually sleep with, for example, the right hemisphere first, which allow the left hemisphere to coordinate all the muscular activity related to swimming and breathing, and allowing them to go to the surface, breathe, while the other hemisphere is sleeping. And then you have the reverse, and hemispheres like this are turning around to be able to sleep. And this is something that is not only seen in dolphins, but also migrating birds have this kind of hemispheric field sleep. So when they have to sleep for days or even weeks when migrating, they will have one sleep hemisphere sleeping at a time. So there are already evidences in the animal kingdom or in some certain pathologies that sleep and wakefulness can sometimes be intermixed. What I will try to show here is that it's not a special case for special individuals or special animal species. We also, to a certain extent, enter this state of sleep-wakes mixtures. And the first thing I'm going to talk about is being awake while asleep. So when you're sleeping, can your brain transiently and locally wake up without any other sign of global awakening? To explore this, I think it's first important to remind what is sleep, because we often It's a common word used by everyone, but in the end, what does it mean to be asleep? And for this, you have actually several potential definitions. The easiest one, and the one that you can always use, for example, in your private life, is asking. So if you want to know if someone is asleep or not, you just have to ask. If the person is not answering, usually it means that they're asleep. So from a behavioral perspective, sleep has always been defined by this state of unresponsiveness. It's important to stress that it's a state of unresponsiveness because it doesn't, sleep doesn't necessarily mean unconsciousness, because when we sleep, we can be dreaming, for example, so we can be very well conscious. It's just that usually we're conscious of something completely different than the environment we're sleeping in. So the prime of behavioral definition of sleep is that you can use it in humans if we trust each other, but how can you ask a fly if she's sleeping, or any other type of situation where communication is an issue? So hopefully we have now other more objective definition of sleep coming from physiology. So we can look at brain activity during sleep, and we can identify markers of sleep activity. So for example, in the transition from wakefulness to sleep, so wake is here and sleep is here, we can observe these changes with brain activity here recorded through an electroencephalogram, show an increase in high amplitude slow oscillation. So this is, by the way, a snapshot of one of the first EEG paper ever published. So from Loomis in 1937. So maybe one of the first ever EEG recording published. And actually, sleep was used to benchmark and to make sure that EEG was a good technique to record brain activity. Because if you have a system that is recording electric activity at the surface of the brain, how can you make sure this is meaningful information? So one way is to take a massive change in terms of behavior or what people experience, and to check how this translates in terms of brain activity. So that's why they use sleep actually to make sure that EEG was working. And EEG is still nowadays, almost 100 years later, used as the best way to define if someone is awake or asleep in science. So with more detailed techniques of recording of brain activity, we can also see how sleep is impacting not only the global electric activity of the brain, but more the behavior of individual neurons. So for example, you can see here what is a classical sleep activity with this high amplitude slow wave that we are going to talk a lot today. This slow wave that you can record on the scalp are often associated with episodes of neuronal silencing. So when you transit from wakefulness to sleep, what happens in your brain is your neurons get more or less synchronized, and they will display this population activity where they will fire in unison and get silence in unison. And that change from a desynchronized activity to a very synchronous and alternation of active and non-active state is really what characterizes sleep. It's also thought to be the reason why we lose consciousness when we fall asleep, or when we enter a dreamless sleep, or why we cannot respond to our environment. It's because the brain, being hyper-synchronized, it cannot do the computation it does when it's awake. So this pattern of activity here, this episode of activation silencing that corresponds to this slow wave, is really thought to be the basic element of sleep physiology. So this is a snapshot of a few seconds of sleep in a patient implanted with depth electrodes. So these patients are epileptic patients that are implanted with electrodes to locate the locus of the epileptic seizure. So they are monitored in the hospital for about a week to pinpoint where the epileptic seizure is, and then they're getting a surgery to take this brain region out. The good thing is sometimes these patients accept that their data is used for other purpose than their own surgery, and in particular for science. So this is a work that I've done a lot with Yuval Nir and the Tononi-Chirley group in Wisconsin to study sleep physiology from both outside the brain, so here with a Scalp Eeg, so the kind of thing you will be able to record in any healthy healthy participant or patient. And then, thanks to this patient, you can have information about what's going on inside the brain. So both about regional activity, so this is the EEG in one specific brain region, so for example, the supplementary motor area, but you have also information about single units, so how neurons work in unison and in relationship with this more global activity. So this snapshot of 20 seconds of sleep is representative to what was classically thought about sleep. So sleep as this, what I just described, this very global hyper-synchronized state. You can see that very different regions, amygdala, hippocampus, internal cortex, supplementary motor areas, all these regions display more or less the same thing. So these waves that are synchronous, and each of these waves correspond to an episode of neuronal silencing that you can see in very, very different regions. So that will be a very striking case of hypersynchrony, because you have regions that are very distant displaying the exact same pattern of activity at the same time. The problem with this global picture of sleep is that it's actually not representative of what's going on in the brain. So in the brain, many times you have this, even in regions that are homologous, so just here the left parietal cortex and right parietal cortex, you can observe a wave in one region and nothing in the other region. Sometimes you can also observe a wave in the scalp, so you can see there's an inversion of polarity that is perfectly normal. So you can see a wave in the scalp that corresponds to a wave in one brain region, but not in another. So you can see here there is no neuronal silencing. Later on, this brain region in turn show a slow wave, but you cannot see neither something here in the left parietal cortex or on the scalp. So it's a case where these slow waves that were thought to be hypersynchronous and global are actually local. And if you detect the waves, this brain wave inside the brain, and look how local or global they are, you can see here that most of them are actually local. So what that means is that this neuronal silencing that is classically observed in association with slow wave is not happening everywhere. In some brain region, you still can maintain potentially processing of information. So that doesn't quite qualify us as close to dolphins, but still goes in this direction that when you sleep, left and right part of the brain can sleep in a different way. So this local modulation of the quantity amplitude of slow wave has actually been interpreted as a consequence of prior brain activity. So in this experiment, for example, they had participants doing a learning task where they had two conditions. So this is a task. So, you have to, it's a motor task where in one condition you're tricked or not. So one condition, the rotation one, is more intense than the other and potentially more tiring. And what they observed, if after the most demanding task, you can observe in the sleep after this task more slow wave and higher amplitude slow wave in brain regions that correspond to the motor cortex. So the message of this very important article published in Nature in 2004 was that if you're overusing one specific brain region, you're building up sleep pressure in that brain region, and that sleep pressure will result in more sleep in that specific brain region. So in a way, the brain will be able to adapt the depth of sleep depending on how much a region has been solicited during the day. But local modulation of sleep is not about this absence or presence of slow wave. In some cases, you can even have signs of local arousal. So it's not like sometimes you have or not presence of sleep marker. In some cases, you can even have the presence of clear markers of wakefulness while the rest of the sleep of the brain is staying asleep. So for example, these are still data recorded in patients so that are implanted. So the bottom line here is what you record from outside the brain, and the two top lines are what you record in the motor cortices or prefrontal cortex. And you can see that in these two cases, so it's two different patients, you have this slow wave that are very, very classical of sleep. So someone who will not have access to information about what's going on inside the brain will say, well, these two patients are asleep. But if you record different brain regions, you can see in these two cases that at some point, one of the brain regions, so here the motor cortex, just woke up for a few seconds. But that was not a global awakening. I mean, both at the scalp level, but other brain region inside the brain, you didn't see such local awakening. So it seems that things are a bit flexible, and it's not only that you can be more or less asleep in different brain regions, but you can even have one brain region waking up and one staying asleep. Another study, this time made in Lyon, showed that in France, showed that you can even have some sort of behavior associated with this local arousal. So in this study, they studied patients where they were interested in pain sensation during sleep. So they were inflicting some painful stimuli to people while sleeping, and these people had to indicate by lifting the finger when they were feeling the pain or not. And they had this one occurrence where you have someone asleep, but locally in the motor cortex, they can see a local awakening, and this local awakening was paired with the patient lifting the finger in response to a stimulus. So from an external perspective, you have no reason to think that this person awoke at any point, but what you can see is one brain region waking up and this leading to some sort of interaction with the environment. So then comes this important question of, is sleep modulation only about what you did the previous day, or the environment in which you sleep? Can it predict also how the different regions of your brain are going to sleep? So exploring this question was a very interesting study published in 2016, where they studied what's classically known in the sleep literature as the first night effect. So the fact that when you sleep in a new environment, you usually have a sleep that is decreased in quality. So why would that be the case? Well, there can be obvious ecological advantages of this. If you sleep in a new environment, you don't know anything about it, that can be dangerous. Maybe it's better to have a lighter sleep than usual to be able to respond to any threats if any comes. What they saw in this study that this lightening of sleep was not uniform across the brain. It was actually mostly the left hemisphere that was sleeping less, as if the left hemisphere was, sacrificing for the right hemisphere and sleeping less than usual to be able to respond to the environment. So once again, something that goes a bit in the direction of the dolphins. What they also showed is that this is not just some kind of unspecific lightening of sleep. The left part of the brain was actually more sensitive to deviant sounds compared to standard sounds during the night. So this modulation of sleep depth can have a functional advantage, allowing you to better process or to be more sensitive to unpredictable events in your environment. So that leads to the research that I did during my PhD, so under the supervision of Sid Quitter. So this is a project that started actually in partnership with Sid Quitter and Tristan Beckenstein in Cambridge, where the idea was to see if sleepers can flexibly process what's going on in their environments. So can they even respond or prepare for a response when they are stimulated? So to explore that, what we did was a quite simple experiment where we had people coming and we asked them to do simple semantic categorization. So you have a series of words coming to you through speakers, and you just have to indicate in that case if the word refers to an object, a knife, table, spoon, or an animal, sparrow, koala, this kind of. So they just had to press right, for example, for an animal and left for an object. What we did was to take students, so chronically sleep deprived and happy about it, and we had them come to the lab, sitting in a very comfortable chair, pretty much like here. So the only thing is the light was off and they had the authorization to fall asleep. So what happens in that case is obviously they do. So what we did is when we could confirm that they were deep enough in sleep, so that represents kind of the staircase toward sleep depth, we continued presenting these stimuli, but this time we presented new ones. So stimuli they never heard, still of the same categories, animal or objects, but new ones. What we were expecting is, since while falling asleep, they did this very monotonous task of answering right or left, depending on the semantic category, we thought maybe their brain would just continue to do so while falling asleep. The advantage of having left and right responses is that it's something you can actually see in the brain. When you're preparing for a right answer, you have, it's your left hemisphere preparing for that motor response, and vice versa. So by comparing left and right hemisphere, we can actually track whether participants are preparing for the correct response. And since the mapping of the response and the word category was completely arbitrary and counterbalanced across participants, if we see differences across left and right hemisphere during sleep to an arbitrary rule, that cannot happen by chance. The only thing that it can mean is that participants are not only sensitive to these sounds, but they can encode it, access its meaning, and apply the rule for preparing left or right response. So they mean that in a way they can process complexity of the environment and almost interact with it. It's just that they were not obviously pressing any button, because that's how we define sleep, so by the absence of response. So what you see when comparing the right and left hemisphere when people are awake and responding is that kind of activity. So starting a bit before the reaction time, you have this asymmetry between right and left hemisphere that is centered on the motor cortex. What we saw in our sleep experiments was something quite similar in light sleep. So over the same spot, so motor areas, and the same kind of asymmetry, it was just much delayed in time. So as if computation were taking longer. But this delay in time is actually not so large. It's actually correspond to the reaction time they were doing when quite tired. So their sleeping brain seems to be as slow as a drowsy one. So obviously, even if we were expecting and looking for this kind of results, it's quite surprising to think that while you sleep, you can still make categorization about words in your environment. So we tried to replicate that result in several experiments, and we actually did it in 3 different ones, recording in 2 different countries on 3 different tasks. So I think it's pretty good evidence that there is something going on here. So the next question was, if sleepers can process this information while they sleep, does it correspond to local modulation of sleep? Is it supported by this local sleep aspect that we have seen before? So if I'm talking about it, obviously it's because it's the case. So this is this signature of a preparation of a response. So you can see it appears on the motor areas about 3-4 seconds. So what happened in the brain when you present a sound is initially the reaction of the brain is to increase synchronization. So this is a time frequency decomposition of the brain reaction. You can see in the low frequency range and what's called the spindle range, you have this massive increase in power, this massive synchronization that is centered in the front of the brain, and that has usually been interpreted as a sleep protection phenomenon. So there's a perturbation in your environment, reaction of the brain is to push you even deeper. What we observe after that is actually a decrease of this synchronization. This decrease was corresponding in time with lateralized readiness potential, so this motor response, but also in space. So it seems that when you receive a sound while you sleep, first reaction of the brain is to push you deeper, but in a second time, actually the parts of the brain that are relevant for the processing of this information seems to sleep in a shallower way, and this shallower sleep seems to be related with your ability to actually process this information. We also looked at whether this local modulation of sleep, this self-regulation of sleep, was associated, sorry for the typo, with the learning of new information during sleep. So I'm not going to present that because it's actually a quite complicated paper. But to break the suspense, yes, not only you can learn during your sleep, but this is associated also with this local sleep. We also try to study the dynamics of this phenomenon. So how dynamically sleep is regulated when there is information coming from the outside. And to do this, we used a technique called stimulus reconstruction, where you can track actually what kind of information participants encode from their brain activity. So this stimulus reconstruction approach has several advantages. The main advantage is that you don't need to probe the brain at regular interval, like classical event-related potential studies, where you present one stimulus at a time. You can actually present normal speech to people, and you just analyze their reaction to continuous speech. So the way it works is you can display that kind of stimulus to participant.
So are the most common methods of river crossings.
So just speech. So this speech was presented to a ferret that was implanted in the auditory cortex. You can record the brain activity of the ferret and try to decode what the ferret heard. And this is what you get. So a bit noisy, but still understandable. And it's even quite surprising since the ferret, of course, doesn't know English. So this kind of thing obviously works well when you can implant animals with electrodes and record directly the activity of their auditory cortex. It can also work in humans if you record activity from the outside. Just that instead of reconstructing something complex that you can even hear and play, what we reconstruct is the envelope of the sound. So fluctuation in the acoustic energy of a signal. Because when I speak, for example, there are modulation in the intensity of the sound that I pronounce. And imagine that you're in a discussion, two people are speaking at the same time. If you focus on one person versus another, your brain will be more sensitive to the fluctuation in the acoustic energy of the person you are listening to versus another one. So if you can track this fluctuation of envelope, you can actually predict who you are listening to by using the brain activity. So this is pretty much what's presented here. You record the EEG. From the EEG, you reconstruct the envelope, and then you compare the envelope with the original signal, and you try to guess, in the case of several people speaking at the same time, who you're attending to. So what we decided to do was to use that kind of technique to see if people can select information from their environment. Because so far we showed that they can access to information in their environment. What we wanted to see if they can select what is most relevant. So we placed sleepers in situations where they are presented to two kinds of information, one that is relevant and one that is not relevant. To make it easy for sleepers to know what's relevant or not, what we use is speech, because we're quite used to speech, and we presented them with two kinds of speech. Either a speech in French, because this experiment was done in Paris, or something that looks like French, that has the same grammar, same phonology as French, but it's just not meaningful. So it just made up words. So the technical term in linguistics is Jabberwocky, in reference to the Lewis Carroll. So basically, our sleepers were listening either to French or to French Jabberwocky, and what we wanted to see is whether they will be attracted to the meaningful stream. Good thing with the Jabberwocky, if you don't know anything about French, you cannot make the difference between the two. They are actually the same. But if you know a bit about French, you will be automatically attracted by this. So just to give you an example, this is English.
Not an idle brain. A lot of information.
There's a bit of a lag. I'll just play it here. Yeah.
The sleeping brain is not an idle brain, but can actually treat a lot of information.
So that will be an equivalent of Jabberwocky of this sentence.
The mere can reas os nad and equarias, but pars woolly for a lot of zelizi.
So now you can play, I will try to play the two in the same time.
Bridges, tunnels, and ferries are the most common methods
The
of river
sleeping brain
crossing.
is not an idle brain, but can actually treat a lot of information.
So now we'll play the two together.
The sleeping brain is not an idle brain, but can actually treat a lot of information.
So for English speakers, which I assume is most of the people in the room, it's actually quite easy to focus on the English, well, the meaningful stream, and ignore the other one. And this is something we do quite automatically. So our question was, does the sleeping brain also do this kind of selection of the information based on its meaning? So that's the overall protocol. So I will just go very quickly over about it. But there was a training part where we built our model used to reconstruct stimuli, and then participants were exposed to either Jabberwocky or real speech in both wakefulness and sleep. And what we did was to reconstruct what they were hearing, so to speak, and compare it to either the meaningful stimulus or the meaningless one, and which one is better reconstructed. What we observed that in wakefulness, we can reconstruct better the meaningful stream, but not in sleep. And this is not trivial, because if you present the two information separately, you actually reconstruct them equally well. If you instruct people to focus on the Jabberwocky, this time you reconstruct the Jabberwocky well. So it's really related to the focus of our attention and not to the nature of the stimulus. And what we show here is even when we are asleep, you have a limited advantage for the meaningful sleep, for the meaningful stimulus. And this was done in all sleep, so both light and deep. These effects are actually even stronger in light sleep. The reason why I'm talking about this is because with this kind of technique, we could actually see how the processing of information was temporally and dynamically regulated during sleep. Because as I told you, during sleep, we have these slow waves occurring from time to time. These slow waves are associated with neuronal silencing. What we observe is during these slow waves, so when locking the stimulus reconstruction to the slow wave, is that we lose the ability to reconstruct the two stimuli during a slow wave. But what's quite interesting, what happens after a slow wave. After a slow wave, not only you reconstruct more than chance, so above zero, the two stimuli, but you start seeing a difference between the meaningful one and the meaningless one. And if each slow wave will both kill whatever processing of information you are doing, but also provide you a short window right after the slow wave where you are a bit more awake and a bit more able to not only process what's going on in your environment, but also select which information is the most important. And I think this is actually quite a neat and beautiful mechanism. You can imagine yourself sleeping in a room. If there is one isolated sound, that's not very interesting. You shouldn't wake up to any isolated sound occurring during sleep. So the good thing with this slow wave is if they are provoked, and they are often provoked by external stimuli, you will just kill the activity associated to this. You won't process isolated sounds. But then if you have a series of sounds, if the first sound is followed by other sounds, like door opening followed by footsteps, then you have a window in which you're actually quite sensitive to what's going on in your environment. So you will potentiate your ability to process information that is quite relevant to you. And this is due because the fact that sleep is this a flexible, self-regulated process. So I will go very quickly on this. So sleep is not a monolithic phenomenon, and the way each version of sleep depends on the prior activity and environmental demand. Now I'm going to talk about the kind of thing I'm doing here, which is the reverse image of this being awake while asleep, which is being asleep while awake. So if we acknowledge that sleep activity depends on prior activity and some brain region can be more tight than the other, does it mean that they can also fall asleep sooner than the other? So this is something that has been actually shown in animals. If you sleep-deprived rat, for example, you can see in wakefulness, this slow wave and neuronal silencing, either local or global, that are quite reminiscent to what happened during sleep. And they are more and more frequent the more you sleep deprive these rats. Very importantly, this slow wave can be associated with behavior. So when they occur in a task, they are usually followed by an error. And this is done in a region-specific fashion. So if a brain region is important to do a task, so here the prefrontal cortex, any slow wave occurring in that region will tend to impact behavior, whereas regions that are not involved in the task don't impact behavior. So it seems that when you are tired, part of the brain can fall asleep. And if one specific region falls asleep, then it will perturb what this specific brain region is supposed to do, and that can have a consequence on the way we behave. So this is something we confirm still with working with Yuval Nir on this implanted patients. So they had to do a simple visual task where they had to do simple face place recognition, and we examined their reaction time. And what we saw is that lapses, so when they get tired, they tend to be slower and slower. These lapses were actually associated with a reduction in the firing of neurons. So neurons themselves, in a way, getting tired. Luckily, this kind of sleep-related activity in wakefulness can also be observed using Scalp Eeg. So in Scalp Eeg, you can actually, from time to time, see this slow delta theta wave that increase in number with tiredness. They also are associated with behavior, with behavioral impairment. So they are a good index of this fatigue and this tendency of fatigue brain to enter mixed state of sleep and wakefulness. So this is actually a project we're doing here at MBI with this student involved. So it was the honors project of Tegame, McKay, to see how this local sleep affect both not only objective performance, but also subjective experience. So we have a task that has been designed to be very, very boring. People do this task, so it's simple go, no go. And we examine both their performance on the task, but also we interrupt them from time to time to ask them what they had in mind. So whether they were focusing on the task, whether they were thinking about something else, or whether they're just mind blanked. And what we observed is that, first of all, they are focused on the task only 50% of the time, which is worrisome, but has been shown in many, many, many papers. What we also observed is in the EEG, we could detect these local sleep events. So this local slow wave. So just for comparison, this is the kind of wave detected in wakefulness. This is the same thing detected in sleep. So it's very, very similar. The only difference is the amplitude. What we also observed in these participants was that the number of these slow waves was positively correlated with fatigue. So this is quite interesting because these people were not sleep deprived. I mean, they're as rested as you are, yet they were showing from time to time this slow wave intrusion during wakefulness, and this was correlating with fatigue. So local sleep in wakefulness doesn't appear like an extreme event. It could be very well something very frequent happening each time we are tired. We could also see that the prevalence of this sleep occurrence was negatively correlated with behavioral efficiency. So there were more slow waves when participants were impulsive or quite slow on the task, and this in an opposite relationship with pupil size. So it seemed that when people get tired, when pupils get smaller, this also corresponds to in the brain with the occurrence of this local slow wave. These local slow waves were predictive of errors, but in a very interesting way, because we also found this regional specificity. What happens is not so much the occurrence of slow waves, but where they occur. For example, when slow waves occur in the front of the brain, this positively correlated with false alarm in impulsive behavior. Well, when it occurs more in the back of the brain, it correlates with the increase in misses or the slowing down of reaction time. So when you get tired, your brain region will tend to more likely enter in a state of local sleep. Which brain region enters this state of local sleep will impact behavior. So in this schematic way. So if it's the front of the brain, you get more impulsive because you lose executive control. If it's the back of the brain, you get more sluggish because you process less your environment. And finally, that will be the last result of this. We also saw that this local slow wave were also predicting the kind of subjective experience people had. So they were more present when people were mind wandering, and even more so when they were mind blanking. And their amplitude was also predictive of the kind of distraction people were experiencing, still respecting this frontal-parietal divide. So when slow waves are bigger in the front of the brain, people have the tendency to mind wander more, as if they were still able to be conscious, but not able to control the stream of their thoughts. When it's up here in the back of the brain, they were more likely to be mind blanking, so not thinking about anything. So we can complete this picture by saying that not only local sleep impact behavior, but it impacts behavior in a region-specific manner that will both impact objective behavior and subjective experience. So to briefly conclude on that part, it seems that sleep deprivation can occur when we're sleep deprived, but not only when we're sleep deprived. So it seems a quite general phenomenon, and this phenomenon has consequences in the way we interact with their environment. So I will finish with this nice quote from William Shakespeare about the importance of our sleep in our everyday life, and how wake time and nighttime can actually profoundly impact each other, and how during the day we can enter this mixed states of wake and sleep, and how this will impact the way we actually interact with our environment. So I will finish by thanking you, the audience, also all the people involved in this project. You can see that it's a network of quite a lot of people, mostly based at Monash in different themes of the School of Psychology, because it's really integrative research that touches to a lot of different topics. But I will mostly also thank now Tsuchiya and all the Tsuchiya lab, because it's where I spend most of my time, and I'm very happy to have joined this team, because I think we're going to do great things. Thanks. Yeah, sure.
Preferably not questions on geography.
Thank you. That was a very riveting talk. I have two questions. One is more of a general interest, and the other is more of an applied question. So with the applied, I was wondering, there are a lot of roles in society that require people to stay awake for really long periods, like doctors, for instance, or people in the army. Do you think there's a way of optimizing the local sleep phenomenon that would allow, say, doctors to task switch in an optimal way so that they are allowing some aspects of their brain to sleep and the others to remain active, and do that in some way that would, reduce errors, reduce risks, etc.?
That's actually a very good question. There is a lot of research done on this, how we can optimize wake and sleep. So how we can optimize wakefulness when we have to do something important, and how we can detect instances where it's better to go and rest. Also approaches within sleep of understanding how we can wake up people in the most efficient way. What are the sleep stages where it's better to wake up people, but also more interventional approaches on sleep. So there's a whole body of research on the boosting of these slow waves. So these slow waves seems tightly linked to the recovery aspects of sleep, and they are sensitive to sounds. So one idea is to play sound in synchrony with these slow waves to try to entrain them, make them bigger, and ultimately make sleep more recovery. And it works to some extent. So you can make these slow waves bigger, and it seems that it reduces a bit the time you need to sleep. The prime is, and the big unknown for this approach is, we don't know the consequences on the long term. So, and knowing how sleep is sensitive, not only for brain function, but for all their physiological function, it's quite risky. So you can maintain people for long periods of time, but that can have terrible consequences in the future. You can maybe try to optimize wake and sleep, so not to keep them awake, but just to see if they can sleep less. But we have no idea of what can be the consequences.
Thank you. And if you don't mind, unless there are pressing questions, I have a very quick question about the function of surprising sounds that are not followed up by important other sounds while you're sleeping. The subjective experience is often that sounds in the environment can be very quickly incorporated into your dreams. Do you think that is sort of the expression of that function? Or
so it's actually, when you look at on the research done on this, it's actually the reverse. Most of sounds are not incorporated into dreams. It's very difficult to incorporate efficiently sounds into dreams. Usually when they're incorporating, they lead to an awakening. That being said, there is so few done, surprisingly, on dream research that maybe it's just that people didn't use the correct simulation or the correct way of doing this. So there are actually a lot of novel interest into this. For example, Now was telling me that he visited a lab in Chicago where they're using lucid dreamers, so people who can communicate while they sleep. And they have examples of people who can actually report hearing things during their dream. So they not only incorporate what's coming from the outside, but they can actually differentiate it from what they are dreaming about. So it might be richer. It might be that a lot of stimuli are actually incorporating, but turned into something else. And then you don't realize that it was incorporating, because just as an alarm clock can turn into bells, maybe lots of time there are things that are transformed and hallucinated during sleep, so we don't figure out that we actually incorporated it. But the state of the literature is more or less that we incorporate very, very few of what's in your environment while we dream.
We actually have a question online. Oh, sorry. Oh, stop.
Yeah. I'll ask for Zoom. So we've got a question from Zoom. Fantastic talk, Dr. Andrillon. Have you looked at the impact of local injury, that is stroke, on local sleep? Are there any compensatory sleep mechanisms that allow the contralational hemisphere to sleep more?
Yeah, I've never heard about anything around this, but that's actually a very interesting question. I think the state of the local sleep literature is more like that. It's at the point of the proof of concept in a way. So the fact that it exists, that it matters, but most of the time it has been done in healthy, good sleeper populations. So now there is actually a lot of opportunities to look at more special cases. So a stroke patient can be an interesting thing. I know that in Sydney they have a project on apnea and local sleep. So does sleep apnea wake up part of the brain more than the other, and how is the consequence of this? Here at Monash, and that's actually part of my NHMRC ECF, we wanted to look at inter-individual variabilities in local sleep, and whether some people do more local sleep while they sleep than others. In particular, people with insomnia or who report not sleeping well. Maybe one of the reasons why they not sleep so well, or why they're sensitive to the environment, is actually because their sleep is more fragmented than good sleepers. So studying these inter-individual differences is actually, I think, the next step in this topic.
I have a question about how many different parts of the brain there might be that can sleep independently. I think in your talk you gave sort of mixed results. One was with finger pain stimulus, where people seem to have motor cortex that was awake whilst they were asleep. But then in the language lateralization task, I think you showed there was no differentiation. So is there a number of brain regions that might be able to be separately asleep in your view?
So I think it's a whole gradient actually of more and more or less and less massive differences. It will be interesting to see how octopuses sleep. I mean, they have lots of different brains in a way. So how these brains sleep will be quite interesting. In terms of mammal brain, there is still a lot of things that are unclear and unknown. Most of the time we study the cortex and cortical regions. So they are fairly well interconnected. So possibilities for things breaking down into 5 different regions, sleeping in 5 different ways, is potentially unlikely to occur. But then you have subcortical nuclei, and a lot of them show activity during sleep that are completely different from the rest of the brain. But this has been insufficiently mapped. There could be also some finer, subtle differences. So recently, the Tononi group published a very striking paper where they showed that during REM sleep, actually the different layers of the cortex are not sleeping in the same way. In some layers you can see slow waves, in some other you don't. And we have no idea why it's the case. So these dynamics is very likely to be far more complicated than the original picture of cortex is sleeping globally. There might be a lot of differences that are due to environment, prior activity, or even anatomy of the brain.
I'm just curious, I suppose it's in part relates to pathology and the like. By and large, sleep onset's discretionary. We choose a moment in time that we're going to, although it isn't invariably, for instance, in the middle of a lecture. But in the instance of, say, people with narcolepsy that seemingly fall asleep without any real warning, do we know much about what's going wrong there?
So I don't know a lot about narcolepsy. Actually, I know that it's quite well studied, and it's one of the issues that have been cracked in a way. So I won't dare say too much about the mechanisms, but yeah, the mechanisms down to the genetic level of narcolepsy have been studied. So that's why they do this sudden transition. Is well understood, but actually that's something I didn't include here. But in the sleep onsets, we can actually see these also regional differences. So not all the brain regions fall asleep at the same time, which can explain sometimes why, for example, we can read a book and wake up in the morning and not forget anything about the last few pages. So it could be because the thalamus is going asleep before the cortex. So we can be, when we fall asleep, in a state where we maintain consciousness, we maintain some sort of awareness of the environment. But all this information is not passed to the hippocampus that has already started to fall asleep, or because there is no transfer of information. So this part is lost. And narcolepsy can be one of these other examples that sleep onsets can vary across individuals in its dynamic or in how sudden it is, and so on.
Okay. Are there any other questions that we have for Thomas? All right then. Well, I think it just remains for us to thank him again. And I'll just do a bit of spruiking, if I may, and remind people that this is a regular event, this seminar, and next month we'll be hearing from some of the people here at MBI involved in the preclinical imaging, which may be of interest to you. And I also been asked to remind everyone that the Convergence Science Network will be having a session here at MBI on the 7th of May, and it's going to be involving now and colleagues and examining the question, who do you think you are? Philosophy, neuroscience and the nature of consciousness. So that, I imagine, would resonate with this audience as it is at the moment. So thanks for your attention, everyone, and please do remember that we have this meeting quite regularly. Thank you very much. Thank you, Thomas. That was great. Excellent. Thanks.